Hi, guys. Thanks so much for joining me tonight. I really appreciate it.
Hi.
Hello.
Thanks for having
For
us.
Absolutely. For students who may be hearing about radiopharmaceuticals for the first time, what is a radiopharmaceutical and why are chemists so important in this field?
Radiopharmaceuticals are a type of radioactive drug that can be used to treat or diagnose cancer. They're generally made up of a few components. We have our radioactive metal, and then we have an organic molecule, what we call a chelator, that kind of acts as a cage and it binds to that radioactive metal. Then we link the chelator radiometal complex to a targeting vector. This is the biological component that can actually seek out and bind to, for example, specific biomarkers, so receptors on like the surface of a cancerous cell, which allows for like site specific delivery of radiation to the affected sites.
I also thought it might be helpful to define the term theranostics and why that's such a big deal in modern medicine.
Theranostic basically is a term that combines the two words therapy and diagnosis. So basically is a drug that combines both modalities and can be used for both imaging and therapy.
Why did your team choose lead isotopes instead of something more familiar like fluorine?
So basically we're working on different radiometals, but in this study we choose lead because it possesses two radioisotopes that can be used for therapy and for imaging. So it's a perfect theranostic isotope. There's lead 212 that can be used for targeted alpha therapy to destroy and treat cancer, and lead 203, which is the diagnostic counterpart that can be used to image the tumor sites, whereas fluorine or technetium cannot be used for therapy, they can be used for imaging. So we decided to work in this theranostic.
Something I read today was like it's like treating what you see. So lead 203 and lead 212, they're a chemically matched theranostic pair. So since they're the same element, we can chelate them with the same organic molecule and conjugate it to the same targeting vector. So the idea is the diagnostic agent will then be able to accurately predict how the therapeutic agent will behave in the body. Marianna was actually mentioning something very interesting this week because lead 212 isn't actually they call it an alpha for alpha therapy, so this is depending on like the radioactive metal that you're going to incorporate into the radiopharmaceutical. So they employ it for targeted alpha therapy, even though it's a beta emitter itself, which is a pretty interesting thing as well. Function of the lead 212.
On the topic of chelators, what happens if it doesn't hold on to a metal tightly enough inside the body? And um, again, for the more general introduction at a high level, why is it critical for radioactive metals?
So the chelator acts as almost like a cage, and it just holds that radioactive metal as it's transported throughout the bloodstream because these are injected intravenously. So then they're circulating through the bloodstream, and then the targeting vector is what allows the drug to accumulate at the actual target, so the cancerous cell. So if the chelator let go of that radioactive metal, then it would go anywhere in the body based on the intrinsic properties of the metal itself. So for lead, it would go to the bones and where else, Marianna?
If you don't have a chelator, basically the radioisotope that you inject will just follow its own biodistribution. So you will not be able to target the cancer cells, but it will go also to healthy tissues or organs. So that's why having the chelator is extremely important. Otherwise, you will not be able to target the cancer sites. There's some examples of drugs that don't use a chelator, like radiometal based radiopharmaceuticals. They don't have a chelator, but they're pretty rare, I would say. So this is like the general strategy, and it's one of the fundamental component of this type of drugs.
And when designing a chelator, what are the main things you're trying to balance? Stability, speed, obviously safety, like you mentioned. Are there any other factors?
So the main things we focus on in chelator development is forming a complex that is both kinetically inert, so resistant to those transmetallation or transchelation dissociation events in vivo, because then that would lead to what we were previously discussing, where the radioactive metal could just go wherever it wanted in the body rather than staying with the drug. And the next thing we want it to be thermodynamically stable. We also want it to efficiently complex that radioactive metal. We want it to work at low concentrations. We also want it to be able to grab onto that radioactive metal at biological or at room temperature, for example, so we could conjugate and attach our chelator radiometal complexes to targeting vectors that are, for example, heat sensitive, or targeting vectors that are heat sensitive, like an antibody. You can't like heat an antibody to 90 degrees Celsius.
And from what I understand, there are already well-known chelators like DOTA. Why wasn't that a good enough option here?
Yeah, so the DOTA and DOTAM chelators, there have been associated with rapid clearance and limited uptake in the actual tumor in vivo studies. So the goal by introducing the more hydrophobic pyridine groups in the chelators that we published, DO2A and DCMC, was to increase the lipophilicity. So hopefully in future in vivo studies, the pharmacokinetics will be altered based on the addition of those groups.
And also, in particular, for lead 212, as Simona mentioned before, the actual therapeutic particle is its decay daughter, bismuth 212, and it's not lead 212 itself. If we're no longer able to trap both the parent and the daughter, what will happen is that we might have the release of the daughter, and this will cause all the side effects that we have mentioned before. So one of the main issue with DOTA and lead-212 is this one, is the release of the daughter from the chelator. And again, that bismuth 212 is the therapeutic one mainly. So if we have that problem, it's that what we were trying to solve with inserting also the pyridine, so enhancing also the stability of the lead 212 bismuth 212 transition.
Yeah, I think that's a good transition in terms of when you started this project, what was the main question you were trying to answer?
Well, there was another chelator previously developed by a previous PhD student, now has their PhD, Brooke McNeil. She had cyclin 4 PY, so there was 4 pyridines on the backbone. And in the in vivo studies, there was like very high liver uptake. So the lipophilicity was increased almost, you'd say, like maybe a bit too much. So then we were trying to move backwards just a bit to try to further tune the hydrophilicity versus the lipophilicity of the chelator. But then there's also other chelators, like a lot of Marianna's chelators in literature are sulfur based chelators, which first were hypothesized to work really well for the lead based off of what we know about the properties of lead. But then once she did all the radio labeling and found that the sulfur donors were less favorable than like a carboxylic acid pendant arm, that also helped us construct these sets of chelators where we were like carboxylic acids work well and the pyridines increase the lipophilicity, so let's try this type of combination.
This might be kind of a tricky question, but how much of this work is creative design versus problem solving around known limitations?
I think it's almost a balance of both, I would say. A large part for sure is creative design, like, for example, developing the new ligands, these new structures. But also, we have a lot of problem solving, like, for example, when we have to optimize reaction conditions or improving radiolabeling efficiency, that's more a problem solving part, I would say. But definitely, it's both.
If you had to explain the difference between two chelators without their structures, how would you describe them?
I was thinking about how to answer this question today, and I honestly couldn't really decide what I wanted to say. Did you have any ideas, Mariana?
It's kind of hard because, like, chemical structures for us are like the alphabet, probably.
Yeah.
So we can't really communicate with no chemical structures.
I mean, that might be a great answer, is that to explain the differences between the two chelators, you really need to get into defining them at a structural level to really understand the
Yeah.
differences.
Yeah. And we base our chelators off of what's called Pearson hard soft acid base theory. So that's what goes into our design of the chelators, where certain metals have properties that are favorable with certain bases. So Lewis bases are the donating atoms, and the Lewis acids are the metals. And then we can form certain ideas based off of what Pearson says certain metals like. But it's always a little bit up in the air too, because with lead it's supposed to be an intermediate Lewis acid, but as we've kind of seen a few times, is it also really likes those harder donors. So it just gives us a platform that we can base our ideas off, and then we work from there by just trying substitution and putting on different types of Lewis bases on the chelator.
This podcast is mostly geared towards undergraduates who are learning about separation science in general, whether it's chromatography, mass spec. How does chromatography show up in this kind of research?
Definitely with the synthesis, I perform manual silica columns. We're always monitoring our reactions by TLC. We characterize our complexes by nuclear magnetic resonance. But the chelators, we usually have to purify them by reverse phase liquid chromatography. So that's our main type of chromatography we use at the purification step to get a nice pure organic molecule that we can then proceed with to see if it will bind a certain radioactive metal.
And I think you may have just answered this. What kinds of questions does chromatography help you answer?
Gives us intel to the purity of our complexes.
But also, we use chromatography to determine if we are actually able to bind the radioactive isotopes. So, once we get the compound,
Right.
and again, as Simona said, we purify by chromatography, we also use chromatography itself to determine the amount of radioactivity that is actually incorporated with the chelator. And we use, it's basically the same of TLC or HPLC, but we have a radiation detector, so those are called radio HPLC or radio TLC. And we use this technique also when we want to evaluate the effective ability of our ligand to incorporate the radioactive isotope.
Yeah, that's a really good
So
point.
it's chromatography is fundamental for us to test the efficacy of our systems.
Yeah, and the systems we use, like the instrument, we can inject, for example, the radioactive isotope, and then it will give us a retention time. And then we'll inject the radiometal plus the chelator when they're bound to one another. And then it would give us a different retention time. And then when we challenge the system to see if it essentially falls apart, when we challenge it with human serum, for example, to mimic an in vivo system in vitro, we can then see, okay, well, is it letting go of the radioactive metal? Is there other metabolites that are being formed? So, yeah, that's a great point. We do use that a lot.
And undergraduates usually learn NMR as a way to confirm structure. In this project, how did NMR help you understand whether your chelators were actually binding the way you hoped?
So we use NMR because we can visualize what's going on. So we can take the NMR, for example, the hydrogen NMR of the free ligand, and then we can form the metal complex, and then we can get the NMR of that and identify spectral changes where we see, for example, an increase in spectral complexity and the ppm values of the peaks because the electron environment is changing once the ligand binds to the metal. The chemical shifts of the peaks also change, moving more downfield or upfield depending on the type of binding. We can also use nuclear magnetic resonance NMR for determining the thermodynamic stability constants, which Marianna uses plotting software from determining the chemical shifts versus pH values, where we can actually just see how the spectra looks at each different pH.
I wanted to add, like, usually students are probably just familiar with proton NMR or carbon NMR, right? But also we are also, let's say, inorganic chemists, and many metals are also NMR active metals. So in the case of lead, you can also do NMR instead of the proton NMR on lead, or there's also other metals that are NMR active, like mercury, cadmium. So there's also all these other, let's say, possibilities of using NMR in our field. And in this case, you directly see the chemical environment around your metal ion, which is very interesting.
What are the steps between making a molecule like this and then it being used in a medical sense, like in a hospital?
So that's a very long process. First of all, we design and synthesize the chelator in our case, and then we tested its ability to bind first the cold metal, so the stable metal, and then the radioactive metal, and then we test the stability in vitro, usually the stability of the complex, like in human serum or biological environment. And if the complex is sufficiently stable, we then attach the complex to a targeting vector, the tumor targeting vector, the part of the radiopharmaceutical that is responsible of the delivery, let's say, of the radiation to the tumor site. And then we have to test this radiopharmaceutical in vitro and in vivo, so in animal models, to assess the biodistribution, so the uptake of the compound in each organs. And if we have developed a drug that is sufficiently stable with a biodistribution profile that we want, which is basically the tumor uptake, right? The idea is just to have the radiopharmaceutical at the tumor or the tumor sites, then these molecules can undergo clinical trials. There's different phase 0, phase 1, phase 2, phase 3, which involve like a larger, progressively larger number of patients. And with these clinical trials, usually we assess the safety, the maximum dose that we can give to the patient, the effectiveness, in general, the efficacy, let's say, of the radiopharmaceutical. And after all these studies, then the molecule could be used in an hospital setting. Complicated, multi-step and complicated process before the approval of the drug.
We went to Sherbrooke, Quebec, actually last year for work, and we went to their hospital, and they actually have something very unique at the hospital where they're doing the development of the radiopharmaceutical, the in vitro testing, the in vivo testing, and do they also do inpatient clinical trials there?
Probably. I don't remember there.
But they have almost all of the available settings to test the radiopharmaceutical from the lab all the way to in vivo, like right at the hospital.
That's amazing.
Yeah.
Now, certainly all of these skills build on each other, but Simona, I was especially interested in hearing from you what skills from undergraduate chemistry were most useful to you in this project.
In this project, my co-op at the particle accelerator definitely prepped me to do my master's degree. I was shadowing a PhD student, Brooke McNeil, and she definitely showed me how to do everything from production of a radioactive metal to how to synthesize a chelator, how to figure out how to bind or how to get the chelator to bind that radioactive metal, so do radio labeling studies. It was definitely hands-on work. I never got to take a radiochemistry course specifically. When I started doing radiochemistry, it was a little bit scary because I really didn't know how it worked. But definitely taking any analytical chemistry course was helpful for learning how to purify my chelators or how to run the what Marianna mentioned previously, the radio HPLC to determine how adding that radioactive metal changes the retention time when we're separating, for example, or trying to figure out if the radio metal was complexed by the chelator. But definitely just a practical answer because I didn't take a nuclear chemistry course.
Well, this might be an interesting follow-up then, as far as did either of you picture to work at this intersection of medicine and chemistry?
For me, I don't know if I ever had a direct idea of what I wanted to do because when I showed up to university, I didn't even know what happened there. Like, I'm from a small town in northern British Columbia, Prince George, and I've never met a scientist. I didn't even know what science was when I arrived at university. So I just tried different co ops and took opportunities that came my way and just kept saying yes. And I kind of just like ended up in this field. This field chose me because I definitely didn't have a direction in mind because I honestly had no clue what I wanted to do when I showed up at university. I was just like, I'm going to enroll in sciences. And then I ended up really liking chemistry.
Well, I suppose even the people who go into it thinking they know what they want to do end up changing to something else.
Yeah.
Yeah, so for me, I wasn't really expecting on working in this field. I think I was always kind of be interested in chemistry and being a chemist. Then I started my master thesis in Italy, where I was involved in this radiopharmaceutical research. And from there, I just keep working on this field and I really love it. So
I would love to hear where both of you are most excited about this general field heading, whether it's in the short term, near future, maybe even 5 to 10 years from now.
I think for me, the most exciting is the potential that we have for personalized medicine. So, as we mentioned, we are working on lead and other so called theranostic isotopes because they can allow these personalized medicine. So, basically, the radiopharmaceuticals can be tailored to target specific cancer types and minimizing the side effect on patient and really having a personalized treatment. So imaging that patient, imaging the tumor sites, and then predicting or a therapy which is really patient based. And I think that with all the advances that we're seeing in radioisotope productions, chelation chemistry, the development of the different tumor targeting vectors, we're moving closer to these kind of treatments where they're not only effective but really customized for each patient. So I think that's really exciting in our field.
Definitely agreed. I also think that a wider implementation of radiopharmaceuticals, that would be super cool to see. They're also trying to develop smaller scale accelerators and implement them in more places so the production of different radioactive metals is becoming more widely available. It'll be super exciting if we can actually start to see the progression from the in vivo to the clinical trials and the actual acceptance of these alpha or beta therapies. And not only because we always see conventionally like PET, so the positron emission tomography like F18, but not only just the diagnostics, but see therapeutic radiopharmaceuticals be implemented in the clinic. Would be really exciting.
Was there anything we missed today that you would like people to know about this research?
This could be a pro and a con, but it's very interdisciplinary.
Yeah.
So in my research and Marianna's research, we get to do everything from synthesis, inorganic chemistry. Like I'm also an X-ray crystallographer, so I grow a lot of crystals. Then we're radiochemists. We radio label the radioactive metals. We also can do cell studies once we conjugate the chelator complex to a targeting vector.
Sometimes we're also physicists when we have to produce the radioisotopes, right? So
Mhm.
there's also all that part like physics, probably engineering too,
Definitely, yeah.
biology, then nuclear medicine. It's a very interdisciplinary field. So it's kind of hard to cover all the different parts, but
Yeah.
Thank you so much for your time tonight, Mariana and Simona. I think this has been a really interesting review because it does touch so many elements. For your next project, I would certainly love to have you back on the podcast. But again, thanks so much for your time tonight.
thank you so
Of
much.
course. Can we take a selfie maybe? I'll just take a
Yeah, absolutely.